It was 1944. The Avery-McLeod-McCarty experiments had demonstrated that DNA, rather than proteins, is a carrier of the genetic information. Several scientists worked towards getting a conclusive evidence for the statements made by the trios, until one, along with his colleague, arrived with the proof of a DNA as a genetic material. DNA, the molecular basis of inheritance. The discovery of a genetic material was one of the most important scientific achievements in the human history. A number of brainstorming sessions were held to know the nature of hereditary material for passing on traits onto generations. Most scientists before the mid-1950s had believed that the genetic information was stored and transmitted through the proteins, but not nucleic acids. Though the Avery group's work appeared to be well supported and endorsed by other researchers, the trio met with a resistance from much of the scientific community. However, the debate was well settled by the results from an experiment conducted by an American bacteriologist and geneticist, Alfred D. Hershey. Alfred D. Hershey was born in Owasso, Michigan in December 4, 1908. He received his Bachelor's of Science in Chemistry at Michigan State University in 1930. In 1934, he received his PhD in bacteriology, taking a position shortly thereafter at the Department of Bacteriology at Washington University in St. Louis. Not many scientists were working on bacteriophage at that time. Two other scientists who came across Hershey's papers, Max Dalberg and Salvador Luria, were collaborating on experiments using bacteriophage. In 1946, working with Dalberg, Hershey discovered that phage can recombine when co-infected into a bacterial host. This led to the new area of phage genetics. As leading researchers in the field of bacteriophage, Dalberg, Luria, and Hershey established the American Phage Group, which had a tremendous influence of bacteriophage research. By applying genetic concepts and developing statistical approaches in the studies of bacteriophage, they were able to shed a new light on the range of unanswered questions with genetics. Their team had made a peculiar observation. When two different strains of bacteriophage have infected the same bacteria, the two viruses may exchange genetic information. This led Hershey to write a letter to Dalberg for accepting Martha Chase as his research partner to carry out the further research. In 1950, they joined to the Department of Genetics in Carnegie Institution of Washington, which is now famous as Cold Spring Harbor Laboratory. In the same institution, they devised an elegant yet powerful experiment that has gone down as one of the greatest molecular biology experiments of all times, often referred to as the Hershey-Chase experiment. It was a sensible hypothesis to believe that proteins with their complicated structures were more likely to be the carriers of the genetic information than the simple DNA molecules. So, initially, the experiments which I and Martha Chase did were intended to prove that proteins were the genetic info carriers. Now, in order to do that, we decided to track the transfer of proteins and DNA between the virus and its host. What we selected, we chose to use was T2 bacteriophage as a vehicle for delivering the genetic material to the bacteria. However, Hershey was about to make a discovery that would turn his own notions on end. The T2 phage, with its simple structure, was perfect for Chase and Hershey to further carry out their research. The phage virus produces by injecting its genetic material into the bacterium, leaving its protein shell attached to the host. Then, through a microscopic takeover, the virus seizes the control of the bacterium's reproductive mechanism and uses them to duplicate itself, destroying the host in the process. Though it was known that the protein shell remained outside the bacterium, we thought it was possible that certain proteins were transferred from the virus to the bacterium upon the attachment. So, to prove that proteins are the carriers of the genetic information, we went on to demonstrate that at least a portion of the phage's protein mass was transferred to the interior of the bacterium. The virus consisted two components, a DNA core and a protein coat. So Hershey and Chase had to run two experiments. In each of them, they labeled one of the components using radioactive tags: radioactive sulfur for protein and a radioactive phosphate for DNA. They led the phage particles to infect the bacteria. They had to examine whether the radioactive signals were detected inside the bacteria or outside. To do so, they had to separate the bacteria from the virus, which they did using boring blender. Tom Anderson, along with his electron microscope, had shown that the phage particles would probably, certainly attach to bacterial cells by their ends of the tails. Now, that we know rather precarious attachment immediately after the infection, and sores are ready to create schifacism. The suspension of the phage and bacteria that might break the attachment off, break off the phage particles from the bacterial cells. And we tried various things that didn't work until we tried this elegant little machine, uh, which was invented to make cocktails, and it worked right away. Once separated, they then centrifuged the samples, separating the bacterial cells and phage particles. When they prepared the virus with sulfur-labeled protein and mixed with the bacteria, they found that the most of the radioactivity remained with the virus and did not enter the bacteria. When they allowed the viruses to replicate and collected the new viruses, less than 1% of protein radioactivity from the viral parent were found in the newly replicated virus, implying that proteins were not being inherited. Shocked by the findings, Hershey Chase decided to perform the test once again, but this time with a different razor molecule. They chose DNA labeled with phosphate and found that most of the radioactive DNA was injected into bacteria from the attacking viruses. Much of this labeled DNA was inherited and passed on to the new generations. At first, the pair Hershey and Chase was inclined to believe that experimental procedures were flawed. They rechecked the experimental design. Hershey was a notoriously cautious researcher. His experiments were always well planned and precisely executed. The results were no mistake, and the import of their work were clear. Hershey and Chase had elicited direct, irrefutable evidence that DNA is the source of genetic material and not protein. The experiment added a major piece of the puzzle of finding what genetic information is made of, which won Hershey the Nobel Prize in Physiology and Medicine in 1969. He shared it with Salvador Luria and Max Delbrück for their discovery concerning the replication mechanism and the genetic structures of viruses. Unfortunately, Martha Chase was not included. However, their experiment is still very known for rooting their genetic research traces. In addition to the findings from work by other group of scientists, Hershey and Chase did many more experiments of their own, described in the same classic 1956 paper, Independent Functions of Viral Protein and Nucleic Acid in the Growth of Bacteriophage. In 1962, Hershey became the director of Genetic Research Unit at Cold Spring Harbor Laboratory. His lab continued to work on bacteriophage, focusing on phage recombinations and genetics. In 1974, Hershey retired. He became a founding member of the World Cultural Council in 1981. He was busy, active, and still learning even in his retirement.
Tom Anderson, with the electron microscope, had shown that phage particles probably, well, certainly attach to bacterial cells by their ends of their tails. Now that would mean a rather precarious attachment immediately after the infection. And so it was our idea to try to create shear forces in a suspension of phage and bacteria that might break that attachment off, break off the phage particles from the bacterial cells. And we tried various things that didn't work until we got this elegant little machine, which was invented, I think, to make cocktails. And it worked right away.